Hai and Assalamualaikum warahmatullahi wabarakatuh, and a very good day to all the panels and everyone who is watching this video. My name is Nur Syasya binti Shahreman and I am a final year Bachelor of Science with Honours Applied Chemistry students of UiTM Perlis. I am supervised by Madam Nur Azira Irma binti Muhammad, and today I will presenting on my final year project which entitled A Review on the Inspection of the Amaryllidaceae alkaloids on the tissue cultures of Narcissus papyraceus, or known as paperwhite daffodils, and Narcissus tazetta, or known as bunch-flowered daffodils. I will presenting on 4 contents, which are introduction, literature review, conclusions, and recommendations. First of all, daffodils are known for their decorative bulbous plants and also commonly designed to the genus Narcissus, which makes Narcissus papyraceus and Narcissus tazetta are included in the Amaryllidaceae family. These Narcissus species are used to the synthesizing the Amaryllidaceae alkaloids. So what are these alkaloids? Some of the examples are galantamine and licarin. And in order to produce quality of plant materials, the in vitro culture technique have been introduced to make the mass production happens. And also, as the plant growth regulator is used to trigger the caligenesis of the Narcissus plants. Now, move on to the problem statements, as there is high demand for natural products in the making of medicines or drug, which is in this case are alkaloids. These alkaloids, galantamine and licarin, usually used in the traditional medicines, specifically galantamine used for the treatment of Alzheimer's disease, while licarin can help to cure cancer disease. Thus, alternative approach using in vitro culture are used for the sustainable production of these alkaloids. Next, the significance of study for this review thesis are the high demand for the Narcissus species plants, which give advantages to the researchers to study the in vitro culture system for the mass production of alkaloids, and further research to investigate the use of oxime, which give effects to the alkaloid production, thus increase the medicine alkaloid content produced. Here, I will emphasize on the objective of this study. Firstly, to review the alkaloid contents in the tissue culture of Narcissus papyraceus and Narcissus tazetta, and second, to suggest the suitable approach to expand the productivity of Amaryllidaceae alkaloids in the species of Narcissus papyraceus and Narcissus tazetta. Now, I will explain the literature review on the biological properties of Amaryllidaceae. Antioxidant activity of the sample extraction taken from the plants of Amaryllidaceae express many biological properties, and from the findings, some biological activities are found in the Amaryllidaceae alkaloids, which are, for instance, are anti-cancer, acetylcholinesterase inhibitory activity, and cytotoxicity activity. As for anti-cancer, this family is mainly used for cancer treatment, such as licarin. First alkaloid in the family of Amaryllidaceae have been tested to have anti-cancer properties. Next, the acetylcholinesterase inhibitory activity. Acetylcholinesterase and butyrylcholinesterase both are needed in cholinergic transmission. These enzymes need to be found to have better effects in the advanced system medicaments of Alzheimer's illness. The properties of cytotoxicity and neurotoxicity have been tested on natter-lily and dwarf nerim plants from the Amaryllidaceae family, and the results shows that the process of extraction from the two flowering plants enhance the morphology and practicality of the specific neuroblastoma cells. This shows that both plants can be used for neuroprotective agents for the patient diagnosed with Parkinson. For in vitro propagation section, high biomass content and production of alkaloid influence the times of the in vitro cultivation and the callus culture's presence. The hairy roots as plants for the in vitro culture shows positive signs in producing galantamine alkaloid, because usually hairy roots of a plant will not produce galantamine alkaloids. The enlargement of the in vitro plants are tested using different plant hormone, and the results can be seen on the number of shoots and callus on the plants produced. Next, glycemine and liquorine concentration are determined using different plant growth regulators influence on the nutrient medium. Plant hormones, pictoram, NAA, and 2,4-D shows good results to influence the alkaloid mass production. Study recorded 96% of calyx formation on the plants with NAA at the 25 micrometer concentration. Thus, auxin NAA is the best plant growth regulators for the calygenesis of Narcissus papyraceus and Narcissus tazetta, while pictoram and 2,4-D auxin is not suitable to use for the alkaloid production for both species of the plant. And now I will explain more on the Amaryllidaceae alkaloids in tissue cultures of both species of plants. The alkaloids present in the plants are glycemine and liquorine. Research shows signs of these alkaloids after extracted sample from the plants are tested using GCMS. Next, glycemine types have the most contents in both species of the plant. This proof will be helping to cure Alzheimer's patients. Liquorine alkaloids presence shows that these two species of plants are suitable to use for medical practices. This is because liquorine contains anti-cancer activity and actively advances in plant growth inhibitor. In a nutshell, I will highlight some of the points that have been discussed in this slide. First, the in vitro culture of Narcissus papyraceus and Narcissus tazetta shows the presence of alkaloids which are glycemine and liquorine. Glycemine and liquorine are proven to have biological properties which are useful for the treatment of Alzheimer's and cancer disease. Moreover, the culture techniques also increase the production of glycemine and liquorine. Plant hormones such as auxin are also proven to influence the in vitro culture calygenesis. And lastly, the in vitro culture techniques are also proven to produce multiple types of shoots, roots, and leaves for both species of the plant. And for recommendations, further studies should be done on the plant cultivation process influenced by the plant growth regulators without the use of nutrient medium such as MS medium or basal medium. Instead, the researchers can use charcoal in the induced process. The study may result in full-grown root system of the plants, and the results also can influence on the increased size of the plant bulbs.